And today I'm going to talk about recycling pathways in Pichia cells for intercellular organelles and big structures like glycogen granules during the times of starvation, which helps Pichia and other cells to survive. And in this talk, by the way, this is my lab on the 8th floor here. Unfortunately, we are the only ones studying Pichia, or even yeast for that matter. So that's why it's very, very nice to be here among like-minded people. And next slide, please. If you talk about recycling in yeast, there are two main pathways. One is macroautophagy, another microautophagy. So one involves autophagosomes as intermediates, double membrane vesicles that sequester intercellular content, and another one happens at the surface of the vacuole through direct invagination. Both of these types of autophagy can be selective. Next slide, please. That's why there is myriad of sub-pathways, right? So both macro and micro autophagy can take up big structures like peroxomes, mitochondria, protein complexes inside the vacuole. I like that slide because it's probably the last time when we could put all of them on the same schematic. There are 15 pathways here, but by now the number probably doubled. And surprisingly, this slide doesn't show neither of the pathways we study because they were discovered after 2007. Lipophagy was discovered just in 2009 and glycophagy in 2011. Next slide, please. But before I talk about glycogen autophagy, I'd like to tell a few words about the glycogen metabolism. So as you can see here, glycogen is synthesized by three enzymes: glycogenin, glycogen synthase, and branching enzyme. And it's degraded by two pathways. One is a cytosolic pathway of phosphorylases on the left by glycogen phosphorylase and the branching enzyme. And another pathway which I'm going to focus on today is a pathway which involves lysosomes and lysosomal alpha-glucosidase on the right. Next slide, please. But of course, it's a little bit more complex than just as shown on a metabolism slide. It's not just glycogen, it's actually a glycoprotein. So the process starts by glycogenin through intermolecular autoglucosylation. So there are two subunits of glycogenin getting together and glucosylating each other. So the first two residues of glucose come from intermolecular glucosylation, and then the enzyme can autoglucosylate itself and put more glucose residues, and eventually glycogen synthase joins and extended further, and then the branching enzyme create alpha-1,6 linkages and transfer some oligomers to the site, and eventually this grows into a bigger structure called glycogen granule and incorporates more proteins, regulating glycogen metabolism. So on average, we have 20 to 30 nanometer structures in cells, but the structure can grow up to 42 nanometers, incorporating up to 12 layers of this polysaccharide. Next slide. Yeah, so how those structures appear in the vacuoles or lysosomes in mammalian cells for their degradation. So for this, it's been proposed that STBD1, starch binding domain 1 protein, recognize glycogen in the cytoplasm and then bridges glycogen granules with autophagic membranes by its interaction with ATG8 family protein, and then autophagosomes will fuse with lysosomes where this lysosomal alpha-glucosidase would degrade glycogen to glucose. Next slide, please. However, so in contrast to liver, where this process takes place and is mediated by STBD1, it's not required for such recycling of glycogen in cardiac and skeletal muscles, STBD1 is expressed but is not involved in recycling of glycogen. That's why it's still not clear is it selective or non-selective. And it's important because in Pompe disease, there is accumulation of glycogen in the lysosomes, which jumps the trafficking. And while enzyme replacement therapy was efficient in liver cells, nothing is available right now for heart and skeletal muscles. That's why if it was selective and there was another receptor, we could use it as a target to ameliorate the disease, because if you block this trafficking of glycogen to the lysosomes, glycogen will be efficiently degraded by phosphorylysis process. So that's why we wanted to develop a simple model system to study this in yeast, and we used Pichia for that. Next slide, please. Why Pichia is good? Because in contrast to Cerevisia, which has each of these enzymes of glycogen biosynthesis in 2 copies due to genome duplication event, in Pichia we have just one glycogen one glycogen synthase. It's really easy to study, and it's an excellent model for studies of autophagy. That's why we developed it as a model for glycogen granules. And here you can see that first we deleted GLG1 glycogenin gene and showed that the deletion mutant has essentially no glycogen formation. It's probably the simplest assay I've ever done with yeast. So you just grow them on YPD plate and expose for a couple of minutes to iodine vapors. And if they synthesize glycogen, they turn brown, and if they don't, they stay white. So the glycogen mutant doesn't have glycogen. Then we placed GFP on glycogenin, showed expression, localization to the cytosol, and the rescue of glycogenin mutant. The GLG1 GFP fusion is functional and can place glucose on itself and synthesize glycogen. So we can use it as a reporter protein for glycogen granules. Next slide, please. So first, we studied what happens to glycogen granules when you induce starvation for nitrogen in SD-N medium. And we found that the glycogen granules are transferred to the vacuole in wild-type strain, in protease deficient strain, SND 1163, but not in AGG1 mutant. So autophagy is involved in trafficking step, in the delivery of glycogen granules to the vacuole. Next slide, please. We could study that also biochemically by Western blot, because when the delivery happens, the GLG1 GFP is degraded in the vacuole, but GFP persists. It's a protein from jellyfish. Pichia has a hard time digesting it in the vacuole, so it persists for a long time, and it's an indicator of a successful trafficking step from the cytoplasm to the vacuole. As you can see, AGG1 is involved in trafficking and degradation, and so are vacuolar proteases. That's why the second pathway of glycogen degradation depends on autophagy and depends on the vacuolar enzymes. Next slide, please. So then we wanted to know if this process is selective or not, and for this we used AGG11 mutant. AGG11 is a scaffold protein which is crucial for selective types of autophagy because it organizes pre-autosomal structure and it binds selective autophagy receptors which bring cargo and then passes those cargoes to the AGG8 on the autophagic membrane. An entirely different mechanism is happening during non-selective autophagy. So that's why it's a good tool to distinguish selective from non-selective. Next slide. And here you can see that in AGG11 mutant, there is normal delivery of glycogen to the vacuole. At 24 hours, we can see it in the vacuole, and statistics confirms that. Next slide, please. So same happens in biochemical assay. We can see processing of GLG1 GFP to GFP in AGG11 cells, suggesting that autophagy of glycogen is AGG11 independent. Next slide. Yeah, so, but can this AGG11 independent autophagy of glycogen be selective? Potentially, yes, because we know a few exceptions to the rule, like lipophagy or proteophagy. Whenever Q5 receptor is involved, it's AGG11 independent. That's why we had to go further and develop new ways to prove if it's selective or non-selective. That's the first reporter we used so far. But now we created additional reporters. Next slide. So the first one is the same GLG1 GFP with a mutation of tyrosine at 212. So this tyrosine is where glycogenin gets glucosylated. So if you mutate this tyrosine to phenylalanine, then basically you stop glycogen synthesis. Next slide. You can see that this tyrosine is conserved from yeast to human. And next slide. Next slide, please. Yes, that's why when you mutate this tyrosine, you essentially make this protein cytosolic. And another cytosolic marker we used is PGK1 GFP, which was established as a marker for non-selective autophagy. So now we can compare how these reporters are delivered and degraded in the vacuole. Next slide. But first we proved that this mutation does lead to deficiency in glycogen synthesis. You can see that this mutated GLG1 GFP cannot support glycogen synthesis. Next slide. And then we studied the delivery of these reporters to the vacuole. Surprisingly, this mutated version of GLG1 GFP goes to the dot-like structures in YPD medium. Next slide. However, when we transfer to SD-N just for half an hour, those dots dissolve. After 30 minutes, it becomes entirely cytosolic, just like other two reporters. Next slide. So that's why we studied all of them after prolonged starvation for nitrogen and found that all of them are delivered to the vacuole with essentially same rate. And we know that PGK1 GFP is a non-selective bulk autophagy. That's why we think that the other two, the one sitting on the glycogen and the other one residing in the cytosol, are also non-selective. Next slide. We could also prove it biochemically. So here you can see that after you mutate this tyrosine on GLG1 GFP, you finally start seeing more GLG1 GFP because you can finally precipitate it with TCA. When it's part of glycogen granules, it's acid soluble, but once it cannot put glucose on itself and that doesn't build glycogen, it can be normally precipitated by TCA just like all other proteins. And for the second and third reporter, we could measure the rate of their delivery to the vacuole, and you can see that the rates are similar, suggesting that this mutated GLG1 GFP cytosolic version is delivered by non-selective autophagy, just like PGK1 GFP. And on the next slide, we compared also the relative amount of GFP normalized to total protein to show that mutated and non-mutated reporter are degraded with the same rate, suggesting that the one sitting on glycogen is also delivered to the vacuole non-selectively. Next slide, please. Yeah, however, you could argue that, okay, so maybe all three of them are delivered to the vacuole in a selective way, so that's why you don't see a difference, right? Because all three are endogenous Pichia proteins, and potentially all three could be recognized by some receptors and efficiently delivered to the vacuole for degradation. So that's why we wanted to develop artificial reporters which would be not covalently bound to glycogen like GLG1. That's why efficiently precipitated by TCA and amenable to both microscopy and biochemistry. And we made use of a domain called CBM20, present in STBD1 protein, the receptor for glycogen in liver cells. This domain can bind glycogen very efficiently. Next slide. And it's been already used in a probe called GISC, which contains GST tag, MIG tag, and CBM20 to basically probe for glycogen in mammalian cells. However, they express this construct first in bacteria, then use this protein and also antibodies to GST and secondary antibodies with either HRP or Texas rat to do either ELISA or immunofluorescence microscopy. So it was pretty complicated. We decided to make it easier. We made it genetically encoded. And we also placed fluorescent proteins to be able to do just live microscopy. Next slide. We designed 4 different constructs. 2 of them have GFP with CBM20, and other 2 have mCherry CBM20. And we express them under 2 different promoters. One is GSY1 promoter. It's a promoter of glycogen synthase. And another one is AGG8 promoter. So as you can see, AGG8 has a lower level of expression, but it catches up under nitrogen starvation to the level of GSY1. And we could show that all 4 constructs are delivered to the vacuole in autophagy-dependent manner on the left. And all 4 constructs can be efficiently processed to either GFP or mCherry under nitrogen starvation conditions on the right. And it depends on autophagy and vacuolar proteases. Next slide, please. And finally, we could use these reporters to see if autophagy of glycogen is selective or non-selective. How did we do that? So we eliminated glycogen by GLG1 deletion. And essentially, if delivery and processing of these reporters was selective, then after eliminating glycogen, you make all of them cytosolic, and efficiency of both delivery and degradation should drop, because non-selective autophagy is less efficient than selective. However, we didn't see it in neither case. So delivery and degradation of all 4 reporters was as efficient as in the presence of glycogen, suggesting that autophagy of glycogen granules is a non-selective process in Pichia pastoris, at least under nitrogen starvation conditions. So with that, I would like to leave you with 2 take-home messages. Next slide. So first of all, Pichia glycogen is delivered to the vacuole by non-selective autophagy under nitrogen starvation conditions. And then, second, we have now tools to monitor and manipulate the synthesis, degradation, and localization of glycogen in Pichia cells. Next slide. With that, I would like to thank my team. There are 2 people who did this work. Next slide. It was started by my first postdoc, Ravinder Kumar, but then most of the work was done by Nina Vijayavanta, the PhD student in the lab, with the help of Pranita Patu, very talented undergraduate student. And the work was funded by NIGMS and also MBD fellowship and research initiation grant from Georgia State University. Thank you very much for your attention.
Taras, I just wanted a clarification of this non-selective autophagy still requiring a specific receptor, right? So in nitrogen starvation, when autophagy is generally activated, what is the role specifically of this STBD1?
Yeah, so STBD1, we don't have STBD1 in Pichia cells. It's a protein present in high eukaryotes. And in human, this protein is expressed across the tissues, but plays a role in autophagy of glycogen only in liver cells. Only in liver cells, it has LIR, LC3 interacting region, and through that it can basically breach glycogen granules with autophagic membranes containing AGG8 family proteins. And the reason we have that in liver cells, in my opinion, is that because in liver cells, beta granules are aggregated together into huge alpha granule structure, which might require selective autophagy for their efficient degradation. But in all other tissues of human body, there are small and very numerous beta granules, and it might not be necessary to have a selective autophagy mechanism for them. They might be just efficiently eliminated by non-selective autophagy as many other small structures in the cell.